Welcome to That Dating Show, your hit game show. Today we have two very special guests with us, Mitosis and Meiosis. Mitosis, please introduce yourself and tell us a little bit about why you would be important in our mystery cell's life.
I'm Mitosis. My name comes from the Greek word mitos, meaning thread. Because cells are constantly dying over me, it is so important that I help them reproduce and divide. I'm important with reproduction, growth and development, and tissue renewal. The offspring are genetically identical diploid cells.
All right, Mitosis, we see how vital the mitotic cell cycle is. Now, Meiosis, can you give us a brief description?
Hi. First thing I wanted to say is I'm a one-way process. I go through one cell at a time and produce 4 haploid daughter cells. I just wanted to say that I am so important for not any kind of reproduction, but sexual reproduction. I can cut your chromosomes in half.
Whoa, 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 this is getting steamy. Okay, Mystery Cell, please ask your first question.
Mitosis, you were talking about these phases. Can you please elaborate?
In any cell's life, you're going to go through phases. First, you will go through prophase. In early prophase, you are going to have opposing feelings forming. Your centrosomes are going to move toward opposite poles because the spindle microtubes are going to come between them. Then we are going to go through prometaphase, where your chromosomes become more condensed. Each of the 2 chromatids of each chromosome will now have kinetochores, forming kinetochore microtubules that will jerk the chromosomes back and forth. Then to work past this point, we will enter metaphase, where the nuclear membrane is completely dissolved. Each sister chromatid becomes attached to the spindle fibers at the centromere. Chromosomes move to the center of the cell and line up with their copy. Now this will be the hardest part of our relationship. I know how much the sister chromatids mean to you together, but in the anaphase of our relationship, I will pull them in opposite directions, so the centromere split towards opposite ends of the cell. Then your chromosomes will arrive at the poles and telophase of our relationship. Your chromosomes are going to be at complete opposite ends, and a nuclear membrane will assemble around each set of chromosomes. I will break down your spindle fibers and begin to stretch out and uncoil your chromosomes as other organelles move to the ends of the 2 diploid cells we will have together.
Okay, well that was very honest. Anything for Meiosis?
Yes, I know that processes like yours tend to want to divide cells like me more than once. Please tell me about this dividing process.
Well, I have to be very honest too. I plan on dividing you twice. In my first division, I am going to separate your homologous chromosomes. In my second division, I am going to separate your sister chromatids. First, we will move into prophase 1 of our relationship. Here, synapse occurs and crossing over may occur unlike mitosis. Chromosomes thicken and detach from the nuclear envelope. Similar to mitosis, the centroids migrate away from one another and the nuclear envelope and nuclei is broken down. Then we will go into the metaphase 1 of our relationship. Here the tetrads, which are pairs of homologous chromosomes, are arranged and aligned on the metaphase plate. Next, at anaphase 1, chromosomes move to opposite poles. Although this phase is similar to mitosis in that microtubules and fibers interact and cause movement, this phase differs from mitosis in that I allow your sister chromatids to remain together when the homologous chromosomes move to the opposite ends of the poles, which are guided by the spindle apparatus. In the beginning of telophase 1, each half of the cell has completed a haploid set of replicated chromosomes. Most of the time I will make it simple for you. Each chromosome will consist of 2 sister chromatids. At the end, we will produce 2 haploid daughter cells with 1/2 the number of chromosomes you have. But this is not quite yet the end. Then we will go on to prophase 2, where I will form the spindle apparatus and later move the chromosomes toward metaphase 2 plate. Then we go along into metaphase 2, where the chromosomes are aligned on the metaphase plate. But here, your 2 sister chromatids are not genetically identical due to the previous metaphase. The kinetochores of the sister chromatids point towards opposite poles. Then right after, I would take you on a ride to anaphase 2. Your sister chromatids completely separate and move towards the opposite poles as individual chromosomes. Then I will take you to the final phase, telophase 2. The nuclei form, chromosomes begin decondensing, and cytokinesis occurs. At the end of telophase 2, we will have 4 daughter cells together. Each will have half the number of chromosomes you have, and each are genetically distinct from you and from each other.
And let's see who she chooses.
Since I am a somatic cell, I go through the G1, S, and G2 phases, which are all part of interphase. G1 is where my organelles are prepared, S is where my DNA is replicated, and G2 is where I prepare to go through mitosis. I spend 90% of my life in interphase, but when I am looking to replicate, I'm going to choose the mitotic cell cycle.
There we have it, folks. Mitosis and our mystery cell, which turned out to be a somatic cell, are the perfect match.